Good morning. This is a video tutorial on how to letter and color your three-dimensional DNA model template, okay, from the DNA model activity. The DNA model activity handout looks like this, of course, and by now you hopefully have gone through and done all of the questions and made your model on a Jamboard that I shared with you on Google Classroom. So you should be through question number 9 before you start the 3D model. If you aren't through question number 9, pause this video and go finish up through question number 9. So on the Jamboard that you made, you put together at least 12 nucleotides. Remember, a nucleotide is a phosphate, a deoxyribose sugar, and a nitrogenous base. And there are 4 different nitrogenous bases, and they go together in a certain fit, right? So this Jamboard that I've made has a total of 14 bases. You could have done more than 12, but it has to have at least 12. You're going to translate the code from this template that you made onto the template that I gave you, and then you're going to color that template, and you're going to fold it in a certain way so it makes a three-dimensional model of DNA. So what do I mean by that? Well, I need to take this sequence of nitrogenous bases and transfer it over to my template. That's what I'm going to do. So first, I need to do a little doctoring with my template. So template looks like this, okay? What I'm going to do first is I'm going to divide each of these columns roughly in half and draw a line down the middle in pen, okay? You're not going to need the whole template, but you're going to need most of it because you have at least 12 nucleotides. So both sides, I'm going to draw a line down the middle, okay, dividing this column in half, okay? Then I'm going to transfer that code onto this thing. Okay, so what's that going to look like? Well, remember my code is these bases: cytosine, guanine, adenine, thymine, and so on, right? So those bases are going to go onto my template. So let me start copying that over. CGAT. All right. Up here, I'm going to start in this corner. First base was cytosine, that's a C. The next base was a guanine, that's a G. The next base was an adenine, that's an A. Then a thymine, okay. Then I need to go back and see. I got ACG on mine. All right, fine. ACG. ACG. All right. What you also noticed on your template is that C always goes with G and A always goes with T. So just like on my Jamboard, on my template, C always goes with G. Okay, G always goes with C. A always goes with T. And so on. So the complementary sequence of bases goes over here. This is going to be a three-dimensional model, so it's going to have 2 sides when we're done, which means then I have to copy this sequence onto this other column also. I'm going to be doing some origami with this. And last but not least, okay, since it's going to be 2-sided all the way over in this other column, I also have to put the letters that were over here, okay? So C goes over here, and G goes over here, and A, and so on. All right, so now what I have is I have my template all lettered in the same sequence as my Jamboard was. Okay, CGATACG, and the other side of that matches up. Now, the next thing to do is color. Remember, our color code said that adenine is orange, thymine is green, etc., etc. So I'm going to color this in, but there's a specific way we color it in. This parallelogram shape is the cytosine, the C. So I'm going to color this in yellow. Cytosine's yellow. I'm going to color this in yellow. Okay. And guanine is purple, G is purple. So I'm going to color in this parallelogram over here, purple. Okay. And I'm doing a bang-up job here. You want to do a better job coloring, which means over here on this side, this guanine is also purple. All right, and this cytosine over here is also yellow, like it was over on this side. Okay. All right, my adenine is orange. And my thymine is green. Right? Notice I'm coloring in these parallelogram shapes. So when I color it, it's going to be all these different, this pattern of 4 colors. Okay? When you've got it colored and all filled in, then you're going to watch a separate video that will show you how to fold into a 3-dimensional model of DNA. So once you get up through question number 9, follow those directions for lettering and coloring your template, and you can move on to the folding part.